Thank you, everybody, for coming to our talks today. I'm going to be talking about an experiment that I ran last year as part of my dissertation research, and I'm in the process of going through the data and sort of figuring out what's happening. First, I'd like to acknowledge some of the people who've been helping me with this project, specifically the members of our lab who helped me with sample collection, and grant and tech support who helped with manufacturing some of the components that I used. And also my funding sources are Acer and part of Kelly's NSF career grant. All right, so let's start off with some background. So we know that low dissolved oxygen can cause some pretty dramatic changes in sediment ecosystems. I have this handy schematic here. Let me see if I can get my laser pointer. There we go. Hopefully you guys can see that. So on this handy schematic, we have the change that happens in sediment systems when you go from high oxygen conditions to low oxygen conditions. So in high oxygen conditions, the sediments are happy. They're this browny rusty color, which means that they're oxygenated. And the oxygen consumption of the sediments, which is can serve as a useful proxy for sediment metabolism, is generally pretty high. But then as the oxygen goes down, we get death. This blackish brownish color, or the blackish grayish color, and less sediment oxygen consumption. Similarly, in oxygenated conditions, the animals that live in the sediment, which we generally define as infauna, are large, deep burrowing, they're happy, they oxygenate the sediment, mixing it and generally enhancing rates of sediment metabolism. But then as the oxygen goes down, those animals can't hack it. They are unable to breathe at low oxygen conditions and will either dramatically change their behavior or just die. So why does this matter? So we know that coastal sediments are the site of really rapid carbon cycling, especially in shallow productive areas like bays and estuaries. And we also know that these areas are disproportionately responsible for global carbon burial, and that they experience intense oxygen variability and hypoxia and anoxia, which can affect those sediment metabolism rates. One of the most notable examples of this is right in our own backyard, the dead zone off of the Louisiana shelf that forms every summer. So in order to understand how carbon is moving throughout coastal ecosystems, we have to understand how hypoxia is affecting these systems. But it's not always as simple as oxygen gets low and everything dies. So one of the really interesting things about low DO is the continuum of responses that we can see from infauna that may have implications for sediment metabolism. So this is a handy schematic from Riedel et al. 2014, who did a survey of animal behavioral responses in declining oxygen conditions all the way down to anoxia. So if you look at the axes here, on the x-axis, we have the duration of low oxygen, and then on the left y-axis, we have the dissolved oxygen level. And then on the right y-axis, we have hydrogen sulfide, concentration of hydrogen sulfide, which is generally produced in very low oxygen conditions and is toxic. So we see in long-term low oxygen that things will die off. We get significant mortality, production of hydrogen sulfide. There's kind of a middle area, which we will call intermittent low oxygen, where we get severe behavioral reactions where oxygen is low for maybe a couple days. But then we have the Diel cycle. And the Diel cycle may be affecting animals sublethally, not causing mortality, not killing them off, but causing changes in their behavior that may drive changes in SOD. And these behavioral changes may be different for different types of infauna. And we know that Diel cycling is really common in these productive shallow waters. So this is an example of summertime oxygen cycling in bottom waters at the Arcos Dauphin Island station from a couple of years ago. So we can see that oxygen is cycling really dramatically, going from this line here at 2 milligrams per liter is what's conventionally considered hypoxia. So we can see that oxygen is getting really low. If we just look at this snippet of time, getting really low down to close to hypoxia and becoming super saturated during the day. So the broad question that this brings up is, how does the Diel cycle affect animal activity, and how might this change sediment metabolism rates? So that's what this project was sort of focusing on in the experiment that I did. So the first question is just to figure out, do infauna even respond to a Diel oxygen cycle? And one way that we can figure this out is by exposing animals in sediments to what I'm going to call an idealized Diel cycle pattern in the lab, and then comparing their activity and behavior at high versus low points of the Diel cycle and seeing if their behavior changes. And then a second order question that we can ask is, does oxygen history matter? And what I mean by this is, because oxygen can change so quickly and dramatically in a Diel cycle, we may expect to see sediments being affected by the recent oxygen pattern in ways we may not predict just by looking at instantaneous DO. And I'll explain more about this. So here we have a few different hypotheses about ways that oxygen history could be affecting animal behavior and sediment oxygen consumption. So here we have what I will consider or call a proportional response, where the dissolved oxygen level is varying along the Diel cycle, and we get a corresponding variation in the amount of animal activity and in the sediment oxygen demand. So they're proportionally varying along the Diel cycle, and then corresponding changes in their behavior or their depth of burrowing, things like that. Alternatively, we could see something like what I'm going to call a gas response. So as the Diel cycle is varying, we see that animals are decreasing their activity at night when oxygen is going away and they're stressed out. But then as oxygen begins to increase again, they essentially overreact to the return of oxygen because they've recently been stressed, and overcompensate for that by dramatically increasing their activity, and then seeing that also reflected in the sediment oxygen demand. Alternatively, we could see something like what I'm going to call a lag response, where along the Diel cycle, we see that decline in the amount of activity. But then as oxygen is returning, because the animals have recently been stressed, it takes them a while to recover. So we see this depression in their activity and in the sediment oxygen demand. So the way that we can figure out if this is happening or differentiate between these different hypotheses is by comparing animal behavior and activity at about the same dissolved oxygen level, but at different points in the Diel cycle. So when DO is rising versus when DO is falling, to see if the recent oxygen conditions, say over the past 6 to 12 hours, have influenced what the animals are doing. So in order to answer these questions, it kind of depends on being able to precisely and repeatedly manipulate dissolved oxygen in the lab on short time scales, which is actually really difficult. It's long been a sticking point for investigations into dissolved oxygen variability. So to do this experiment, I actually built this method for manipulating dissolved oxygen. It's a simple low-cost method, and it's a closed-loop system that's based on the sediment reactor system devised by Long et al. 2008 that uses sediment to draw down oxygen instead of what's conventionally used, which is nitrogen gas. So I'll just walk you through what's happening here. So we have an experimental tank where the replicate animals are being held. And in the bottom of this tank, there's a bunch of mud that is drawing down the oxygen or consuming the oxygen in the experimental tank. And then we have a Arduino and the electronic guts of the system in this box connected to an oxygen probe that's sensing the oxygen in this tank. And when it senses that the oxygen is too low, it will open a valve and let in oxygenated water from a reservoir tank. And in this way, it can pretty precisely control the oxygen concentration and manipulate it into a prescribed pattern that you program into the Arduino. So I built this system and I set it up in the lab. We have the reservoir tank here with a bubbler, our experimental tank, the Arduino control system. If these electronics look janky, that's because they are well spotted, but they work, and that's what matters. So here's an example of one of the replicate trials that I ran for the experiment, just to show you what the system is capable of doing. So here on the x-axis, we have the time elapsed in hours, and on the y-axis, we have the dissolved oxygen concentration. So the pattern that I chose was 6 hours of high dissolved oxygen, and then it goes through 2 Diel cycles and then declines down to hypoxia, which is 2 milligrams per liter, and holds that for 6 hours. And altogether, the pattern is 72 hours or 3 days. So the black in this graph is the programmed dissolved oxygen, what I told the system I wanted it to do. And the blue is the average DO, the localized average DO, essentially what the system was actually sensing in the tank. And the system is actually pretty good at manipulating the oxygen. It struggles a little bit with the downward slope because it's depending very heavily on the oxygen consumption of the sediments. But overall, it does a pretty good job. So for the actual study, I used 3 different organisms. So the first was a brittle star, which these animals will burrow several centimeters down into the sediment, and they'll extend their arms above the surface and then transport sediment between the surface and depth. I also used a polychaete worm, Owenia, that has, it builds a vertical tube out of these shingled little bits of shell, and it will feed and also respire with this kind of frilly crown that it can retract into its tube. And then I also used a burrowing clam that will bury its shell pretty deep into the sediment, but it will ventilate and also feed using a siphon that it can extend and probe around in the sediment and also along the surface. So I picked these animals because they are all nominally in the same functional group of deposit/suspension feeders, but they have very different lifestyles. So I can just show you some of the videos that I collected during this experiment to give you an idea of what I mean. So first we have the Brittle Star, which you can see also know that these videos are time lapses of pictures that are taken every 1 minute, which is why they seem like they're moving really fast. But you can see that these the Brittle Star is actually moving quite a bit of sediment, and it's transporting sediment and forming these gallery burrows that are exposing deeper sediments to oxygenated water, which theoretically could be increasing sediment oxygen demand. So the worm, it's a little bit fuzzy, but the worm's crown is right up here. So you can see that it can be highly active in clearing sediment locally around its burrow. And right there, that was a poop pellet that it ejected. Poop is pink because I had fluorescent particles on the surface of the sediment for another part of this experiment. But the activity of these worms could also be contributing significantly to sediment oxygen demand. And then the clam we have here on the right, you won't see the clam itself because its shell is buried, but you can actually see the surface right here of the sediment pulsing as it's ventilating. So this ventilation could be injecting oxygenated water deeper in sediments and contributing to greater SOD. So theoretically, these are the reasons why these animals might be showing an effect on sediment oxygen demand within the sediment. So for the actual experiment, I put these critters into what I'm going to call ant farms, which are just thin aquaria filled with sediment, and allowed them to burrow. And then I placed those into the experimental tank and then manipulated the dissolved oxygen into the Diel cycle. And then I observed and recorded the animal behaviors. Here I have set up a few GoPros staring through the side of the tank to produce those videos that you just saw, all throughout the trial of the Diel cycle pattern. And I also measured sediment oxygen demand with metabolism caps. So this is an example of a cap, and it's kind of hard to see because it's clear, but these were placed on top of the ant farms with a dissolved oxygen probe, and the oxygen concentration decline was recorded over time. And I did this 4 times. So there were 4 trials, 4 runs of the Diel cycle pattern with 1 replicate of each treatment in each trial. So 16 altogether. So what I'm just mostly going to be talking about is the SOD part, because that's what I've been focusing on doing the data processing and analysis for. All right, so starting out, we have the oxygen manipulation of all 4 trials together, where this is the same kind of setup where we have the elapsed time in hours on the x-axis and the dissolved oxygen on the y-axis, on the left y-axis. Generally doing a pretty good job. We have all 4 of these patterns overlain. Again, it's tending to struggle on the downward slope, but this is a pretty difficult pattern to follow. So it's doing pretty well. I also have here an axis that's going to become relevant in a moment that I'll explain. So this is the SOD axis. And what you need to know about this is that more negative SOD here means more oxygen flux into the sediment. So more negative equals greater flux. So I took SOD measurements at all of these different time points. And if we're thinking about what general pattern of SOD we should expect from these data, it should theoretically look something like this, where the oxygen seems to be varying inversely with the pattern where the sediment oxygen demand is very high. It's very negative when the oxygen is high. and the sediment oxygen demand is very low or less negative when the dissolved oxygen is low. So I'm going to start overlaying the data on top of this so you can start to see the pattern. So here we have the control replicates, and they look like they're more or less following that expected pattern. These are the brittle stars on top of those. They are the red stars. And then we have the Owenia, which are the blue squares. And then finally, the clams on top of that, which are the green diamonds. So what we can first see is that the overall trend of the pattern is that it seems to make sense. We expect that SOD is going to decrease when dissolved oxygen decreases. Another thing to note, if you're familiar with SOD as a measure of oxygen consumption, is that this axis is really big. These SOD values are large, like suspiciously large. And at first I thought that this could be a calculation or a conversion error that I had made somewhere along the way, but I went back and double checked and it all seemed to be right. So what I think what's more likely in this case is that it's an artifact of the small volume of the overlying water in those chambers. So I used pretty tiny chambers, which means that there's a very large sediment surface area to water volume ratio. And diffusive flux would likely be faster across that short height of the chamber. So that would also result in a greater magnitude of flux. And since the overall pattern does seem to make sense, for now, I'm operating under the premise that it's more of a matter of scaling and that these values are comparable to each other, but will likely not be directly comparable to other values, say in the literature, that are using much larger setups. Another thing that you'll notice is that there's quite a lot of spread at some of these time points. And if we take a look at the relationship between the dissolved oxygen concentration at the point that these measurements were taken and the measurements themselves, the SOD, we can see, well, first of all, they kind of break out into these different groups because I measured DO at these discrete levels. So I have a mini version of that plot from the previous slide up here where the dissolved oxygen concentrations that I took these measurements at are highlighted and correspond to each of these clumps of data points. So that part makes sense. But clearly there's also a lot of variability in the SOD at each level, and there is more variability at the higher DO concentration that you have, which could complicate comparing between the different dissolved oxygen levels. So I'm still processing these data. I just started the analysis, but we can start to kind of get an idea of what's going on by examining a couple of examples. So to have an example of question 1 of do fauna respond to the Diel cycle, we can look at 2 different time points and compare them. So a minimum and maximum oxygen level time point. So here we have when oxygen is at its maximum, these points, and then when oxygen is at one of its minimums, these points here. So again, one of the problems is that there's a lot of variability within each of these treatments, including in the controls. So in order to help correct for some of this, we can find the difference between SOD for each replicate of each treatment to help control for the variability and try and see what's going on. So taking the difference between the points between the same replicate at each of these time points. So we look at just those 2 time points in the comparison. So this is a plot of the differences between SOD for each replicate at high DO and low DO. So on this plot, if SOD is different at high DO than at low DO, we would expect the boxes and whiskers to be significantly different from 0. So this would be, there's no difference in SOD between high and low DO. So that is supported for everyone except for the clam, weirdly. So SOD of all the treatments except for the clam treatment, which was marginally not significant, showed that there is a change in SOD due to low dissolved oxygen. So then the next question, do infauna respond to the Diel cycle? If they are responding to the Diel cycle and affecting SOD in some special way beyond how SOD is normally varying with DO, we would expect the boxes for the animals to be different from the control box. And that was not supported between these 2 points. So the fauna didn't seem to be affecting the change in SOD in any extra way. So as for the other question, we can extract a couple points and also look at an example of those. So remember whether oxygen history matters. So if you'll recall, we do this by comparing SOD at the same dissolved oxygen concentration on the rising and the falling sides of the Diel cycle to see if there's a difference between the two. So that's what that is. So this is going to look like a similar plot as that I just showed you, but I want to set this up with those hypotheses that I talked about before. So here on this plot, if we were to see a proportional response, we would expect that the data would fall more around 0. So a deviation from 0 would indicate that SOD is different between those two time points, and that would mean that the oxygen history is affecting SOD. So if it falls into the negative difference, then it would be a gas response. And if it was a positive difference, in this case, it would be a lag response. So when we actually look at the data, at first glance, it looks like there could be a pattern, but with a t-test, it just showed that there was no significant difference from 0, again, except for the clam. What's going on there? And it also, when we compare the animal treatments to the control, we see that none of the fauna were statistically different from the control in the amount of change in SOD between those two time points. So here it seems to be indicating that a proportional response is most likely and that the animals aren't having an effect. So what's going on here? Well, first of all, the variability is just very high. SOD data is highly variable in the best of circumstances. Even more problems when there is DO variability and when animals are involved. There's also, as I said before, a lot of variability in the variability, which can mess with simpler statistical tests, which assume that the variance is normally distributed. A first look at these data suggests that they might be exhibiting a proportional response, although again, there is much more analysis that I need to do. But it could just be in this case that animal effects are drowned out by the magnitude and the variability of the sediments themselves and their oxygen consumption, which may be why we're not seeing a really obvious faunal effect at this point. So the next steps for these data are more analysis. There are many more time points than just the ones I used as an example, and there's a ton of information packed into this data set, some of which I haven't even touched on. For example, there could be some really interesting comparisons between similar DOs at high versus low, and between the different animals, and also between the Diel cycles. So if you'll recall, because I ran 2 Diel cycles here for each trial, I essentially have duplicates for each trial that I ran. So it would be really interesting to compare the first Diel cycle to the second to see if oxygen history is important on that longer time scale. It could also be really helpful to compare SOD to the videos of the animals' behaviors and compare those side by side, because these animals will have cycles of behavior that they go through at a certain periodicity. Like for just as an example for the Brittle Star, I have this video again. You'll see at the beginning, the Brittle Star is moving a lot. It's transporting quite a bit of sediment, but then sometimes it's just like straight chilling. So it would be interesting to look at the activity that they're actually doing when I took the SOD measurements to figure out, did I take those measurements in the middle of the infaunal version of a sprint or a nap? Because that may help explain the variability in some of the more outliery looking points. All right, thank you for, I guess, paying attention. I'll leave you with some cool videos of animals doing weird things, and I'll take any questions if you have them.
Thanks, Kara. Does anybody have any questions for Kara?
Hey, Kara, this is John Ryder. Nice presentation. I'm just wondering if, do you think you would have seen a bigger response maybe if you would have let it cycle a little bit lower than 2? Do you think that maybe 2 just wasn't, low enough?
Yeah, I think that's definitely a possibility. I also think maybe if I had let it cycle for longer, I might have seen some responses, because they do seem to have, just looking at the videos and how they played out over time, they do seem to have some sort of response to low oxygen, but it's not always clear exactly where a threshold for that activity might be, if it's at 2 milligrams per liter, or if it might actually be below. So if I were to do this experiment again, I might want it to go lower than that. But as you saw, the oxygen manipulation setup sometimes does struggle with getting really, really low. So I would be a little bit worried about trying to get it to bottom out. And also the reason that I set up the Diel cycle that way is because I wanted it to more or less approximate the dissolved oxygen variability that you see out in the world on a daily scale. And from what I gathered from the monitoring data from my Mobile Bay from those stations, is that it varies in how low it will get, but it's pretty rare for it to completely bottom out on the Diel scale to say anoxia. So I was aiming for hypoxia for that reason. But that is a good point. It could be that they're just not responding because that's not what their threshold is.
Nice talk.
Thank you.
Hey, Kara. Do you expect these the results that you got to be conserved for animals that are burrowing in different types of sediment? So things that may have larger grain sizes, greater porosity, et cetera.
Yeah, that's a good point. So I would expect the simple answer is it would depend. For the sediment that I used was fairly muddy in comparison to what you just said. So you would expect the SOD to generally be higher than sand. But also in sand, you get different animals that have different lifestyles and different functional traits. So it would depend on the animal and the sediment and the combination of the two. There's also the consideration that in sandy sediments, irrigation would be different because of porosity and because in larger grain sediments, advective flow is more of an issue, and permeance of oxygen is easier, so you might see more flux in that way. But as far as sediment oxygen consumption, you just generally expect it to be higher in muds.
Awesome, sounds good.
Hey, Kara, this is Dottie.
Hey.
Really nice talk. I really like this. Really cool. I really appreciate worms when I hear you talk about them. So I have a question about how you could maybe remove the variability from the SOD from the sediment. Could you find a different medium to put the organisms in so that you're just isolating their oxygen demand?
Yeah.
And then you know what I mean?
Yeah. So there have been a couple studies that have done that, where it's trying to isolate the oxygen consumption of literally just the organism itself. One of the problems with that is because these animals naturally exist in sediment, which is a very specific environment, and it would be difficult to say that brittle stars outside of sediment would be behaving in a way that you could reliably say it would if it were in its natural habitat. So you could do that, but I would be suspicious about the reliability of those data because you're taking them out of sediment. Does that make sense?
Yeah, absolutely. Yeah, I would think there would be a lot of caveats, but just thinking of ways to reduce any variability and taking them with a grain of salt, if you will.
Yeah, grain.
Yeah.
We got a couple more questions. Niels, do you want to go ahead or?
Yes.
Niels, hi.
Hello from Long Island.
Hi.
Thanks, Kelly, for letting me know, and thanks, Kara, for a nice presentation. Obviously, I really like this work. I had a question. So first of all, what was the density of these organisms in your ant farms? There was just one individual?
Yeah.
One individual, okay. So that
individual.
could not explain maybe the very high oxygen fluxes to the sediment.
Yeah.
And then you talked a little bit about the oxygen history, let's say. To what extent do these animals that you collected experience diel cycling hypoxia, or have they experienced diel cycling hypoxia in the field? I was a little late. Maybe you mentioned that. And to what extent, I know it's hard, but sometimes I feel like when we bring in animals, we basically start the experiment when we think we are ready and not necessarily in sync with what the animal experience it so live. So I wonder,
Yeah.
did you look for that? If there's maybe it's off phase. So basically, I don't know, the animals get really confused.
Yeah, so that's it's really interesting that you say that, because I did think a lot when I was developing this experiment about how long I should run it for and how many Diel cycles I should let them, go through, because theoretically, if they're out in nature and this is just on a day-to-day basis, the oxygen is getting super low. I'm already messing with that when I'm taking them out of the field, keeping them in a high oxygen environment persistently until I'm ready to expose them. And then I'm only doing 2 Diel cycles, which is one of the reasons why I think it would be really interesting to compare the first Diel cycle to the second to see if there's anything different there. Really, it was just a logistical thing. This was a really intensive experiment to run, and 2 Diel cycles was honestly as much as I could manage. But that's definitely a consideration. Yeah.
Preston, and then we've got a couple questions in the chat. Yeah, so go ahead, Preston.
Hey, Kara, that was really a cool experiment and really nice talk. And I probably have questions that can go way beyond the few minutes we have here, and maybe we can talk with Kelly offline and probably Kevin, my colleague at UT. But quickly, just maybe you said it, I'm sorry if I missed it. Did you vary the lighting as well to simulate daylight and nighttime in your cycles?
Yeah, I didn't vary the lighting because I wanted to, well, first of all, was because I had videos and the video setup was kind of tricky to get right with trying to get the lighting correct. I kind of just had to leave the lights on the whole time. And I didn't want to vary the light and the oxygen, because then, how do you know what's what's actually changing their behavior in the SOD? Yeah.
No, that's understandable. Control one thing at a time. I guess there's some evidence from acoustic reflection measurements that during the sunny parts of the day, that any plant material can generate enough oxygen to make free bubbles in sediment and then change properties that way. So that kind of leads me to my next question. Did you ever see any free bubbles? Were you ever putting enough oxygen in to make saturation conditions and then free gas in the sediment?
According to the dissolved oxygen concentration and the salinity and temperature of the water, it was not super saturated. The bulk oxygen in the experimental tank wasn't super saturated. That being said, I did use mud for the bottom of the experimental tank in the setup to suck down the oxygen. I did use very, very organic rich mud. Like from a marsh in our backyard, like really organic rich, because I really needed it to suck down the oxygen. And I did see what seemed to be some sort of biofilm on that mud at different points during the experiment. I'm not sure what that would have done to the, I guess, the chemistry of the water. I didn't see any like free bubbles being formed from, say, microalgae or anything like that. But clearly having mud in the bottom of that tank is going to change the water chemistry. And that's definitely a consideration.
Okay, cool. Thank you. One last one. This is easy. On the picture you're showing on the far right with the clam, seeing the sediment surface go up and down like that is a really interesting feature. And
Yeah.
What is the periodicity of that motion? Is that...
Yeah, that's a good question. I didn't see this, that they were doing this until I looked at these videos after the experiment. I was like, oh my gosh. Because like I didn't see it when I was, directly looking at the sediment surface during the experiment. And these videos are time lapses of pictures taken 1 minute apart. So, I mean, they seem to be going up and down, once every frame or every other frame. So something like that. Bivalves, not this bivalve specifically, but other bivalves have been known to have pedal gape ejection of water. So every so often they'll just like squirt out some water, and that's what I think might be happening. Don't quote me on that. But it'd be really interesting to look into the regularity with which they do this and if that corresponds to the SOD.
Okay, thanks a lot.
So So we've got a few questions in the chat, and then we'll move on to Cy's talk. So Tina asks, have you explored how long your mud banks that you use to draw down oxygen can last before they no longer effectively draw down oxygen?
Quantitatively, no. Qualitatively, many times while I was developing the system. It seems like after perhaps a week and a half to 2 weeks, the mud is, in my head, I just started calling it like, oh, the mud is tired. It just wouldn't do anything anymore. So then I had to replace the mud in the bottom of the tank. And then it would be able to change the oxygen again.
Cool. And then the next question is from Steve. Have you calculated the phase lag time between the oxygen and the SOD? I'm not sure if you have the resolution to.
Yeah, I don't, it would be cool if I could do that, because then if the cycle was actually shifted a little bit, that would be a pretty interesting thing. But I'm not sure, given the time points that I used and the data that I collected. But if you think that's possible, like, hit me up. I would definitely be interested in exploring that.
That might be more possible with the next experiment.
Yeah. Mm-hmm.
So then the next question is from Janet. As a follow-up to John's question, do you know if all three of these species you tested have similar low DO tolerances in the field? Do you think a more sensitive organism, perhaps a burrowing crustacean, may exhibit more sensitivity to the 2 milligrams per liter?
Oh, is that okay? I didn't catch part of that question. Can you say the second part again?
So do you know if all three of these species you tested have similar DO tolerances in the field? And do you think a more sensitive organism like a crustacean might respond better to
okay. Yeah. Yeah, I definitely think that's possible. So I think the fact that these animals are found in areas where oxygen can cycle really dramatically is itself an indication of their tolerance to some extent, that we can find them and they're alive and they seem to be doing fine when the oxygen is varying. So maybe looking at an organism that is more sensitive, that maybe is not experiencing these kinds of cyclings or as dramatic of Diel cycling, that could definitely be an interesting thing.
And one last question, and we'll move on to Cy. So Neil says, with mud being the oxygen consuming agent, are you concerned about or have you measured sulfide or ammonia fluxing out and impacting your experiments?
Ooh, yes, interesting question. So in the last trial that I ran, I collected water samples. I have not analyzed them yet, but I was definitely thinking about that, especially when I saw, like, a whitish biofilm on the mud in the bottom of the experimental tank. I was like, ooh. So I'm gonna collect some or analyze those water samples to see if there was any buildup of nitrate or sulfide that could have been affecting the animals in the mud. Definitely, that's a good point.
Cool. So thank you all. That was a lot of more questions than I expected, but those were really, really good questions. So thanks everybody. So let's move on to Cy's talk.
All righty. Great job, Kara.
Thank you.
All right, can you share? Okay, so Cy is going to be presenting on temporal changes to coastal muddy sediment structure following resuspension. So go ahead, Cy.
All right, thanks, Kelly. So before I begin, I would like to go ahead and acknowledge funding provided by the Office of Naval Research and the National Science Foundation, in addition to a lot of help on this project from sediment ecology lab members, DISL tech support, vessel captains, and several faculty members here. So shallow river influenced coastal muds, such as here on the Alabama coast, receive and retain the majority of terrestrially derived sediment compared to other parts of the ocean, which makes these sediments especially important for global nutrient cycling and organic matter processing, despite their small aerial cover. So our sediments are also strongly influenced by frequent storms and the occasional hurricane, especially with last year's particularly active season with both Sally and Zeta pretty much directly affecting us. So these physical disturbances can cause major sediment resuspension, often turning our coastal waters into something resembling chocolate milk, which you probably saw if you were driving over the bridge yesterday or today. This disturbance impacts sediment biogeochemical and physical structure. And with hurricane intensity expected to increase due to global warming, coastal sediments may experience heavier disturbance in the future. So it would be especially important to understand how quickly disturbed muddy sediment re-strengthens, not only from a biogeochemical cycling standpoint, but also for coastal engineering applications such as sediment diversions, channel building, and dredge disposal. So most geological work on muddy sediment disturbance focuses on the physical, where small grains get picked up and washed away, while larger grains may kind of move a little bit, but generally stay put. But cohesive muds are not simply these grains packed together, but consist of fine mineral grains kind of embedded in this sticky organic matrix. And this organic matrix is made up of this stuff called exopolymeric substances, which I'll refer to as EPS because it's way shorter to say. And that's a sticky carbohydrate slime that is exuded by microbes and microalgae in the sediment. So EPS produced by abundant diatoms in biofilms has been shown to reduce erosion of surface sediments, but little work has really examined the role surface or subsurface EPS plays in physically disturbed mud re-strengthening, which may be especially important for subtidal muds in turbid waters that aren't going to really have well-developed microalgal biofilms. Additionally, most muds provide a home to burrowing animals known as infauna, and infauna can have a number of impacts on the sediment. So they can loosen compact sediment through their burrowing and feeding, like Kara showed in some great videos, but they can also increase sediment stability by enhancing microbial growth, producing their own mucus or compacting their burrow walls. But how infauna affect sediment stability is highly variable. It depends a lot on the sediment type, the animal's body size, what kind of burrowing it does, and many other factors. So disturbance to these animals' mud home can kill some infauna, but also just destroy their homes. But we really don't know much about how these surviving infauna affect recently disturbed mud structuring by rebuilding their homes, especially since there is no worm FEMA to help them out. So our study focused on how different geological and geotechnical properties of natural cohesive sediments changed over time following disturbance, and what role biological processes play in that sediment re-strengthening. So we expected that the sediment physical compaction, so just that physical grain packing, would occur rapidly and stabilize faster than overall cohesion, which is that stickiness of the sediment. And we expected that surface and subsurface cohesion to increase over time more gradually and on similar timescales due to both depending on microbial EPS production in addition to physical consolidation. We were also interested in the timing of this expected lag between compaction and cohesion stabilizing. So we collected sediment cores from muddy sediments at about 10 meters depth offshore of Dauphin Island using this multi-core. And we used a modified turkey baster with a little prong with holes on the end to resuspend the top 5 centimeters of each core every hour for 12 hours. And the time and depth were chosen because 12 hours is on the order of hurricane timescale passages over the coast. And the depth is half the global average biologically mixed layer and within range of hurricane mixing depths. So it would likely affect animals living near the surface more than larger animals that can burrow deeper and may survive and come back to affect these sediment properties. So we sampled 4 cores daily for the first 3 days and then weekly out to 1 month post-suspension. So just to reiterate, we expected that changes to properties related to mud cohesion would continue to occur after sediment compaction was stable. And sediment compacting over time is basically just the increasing sediment density due to settling sediment weight. To quantify this, we measured the sediment-water interface height, which we'll refer to as the SWI there, relative to the undisturbed height. And this provides a nice continuous and non-destructive metric of short and long-term compaction. And we expected that decreasing sediment-water interface height would directly show this compaction. We also looked at sound speed through sediment at different depths, and this provides yet another non-destructive measurement. So this graph here is the ratio of sediment to water sound speeds on the y-axis versus sediment porosity, which is the ratio of water volume to sediment volume. So the more water in the sediment, the higher the porosity and the less dense it is. And overall, in most sediments, decreasing porosity results in faster sound speed in sediment relative to sound speed in water. So we expected that sound speed ratio would increase over time as sediment compacted. And finally, we collected a subsample core out of the bigger core and sliced it into 1 centimeter increments and dried it to get a measure of porosity, which we expected to decrease with increasing compaction. So now we'll take a look at our compaction results first. So here we have average sediment-water interface height relative to pre-disturbance height in centimeters on the y-axis over time following disturbance on the x-axis, where greater than 0 means it's less compact and 0 is that original sediment-water interface height that is undisturbed. So we can see that average sediment-water interface height dropped rapidly over the first day and then continued to drop slightly from day 2 through day 7. But by day 7, there really wasn't much change the rest of the time. So if we compare porosity, which is now on this right y-axis over time within the disturbed layer, so these points are the averages of the top 5 centimeters, it didn't really capture this rapid compaction and then gradual consolidation over the first few days. There's really not much change in time there. We observed a slightly different pattern in our sound speed measurements. So here we have average sound speed ratio plotted against time, and a reminder that higher sound speed ratio means faster sound, which generally indicates increasing density. And these filled circles are the individual time point scans, while the open circles are the day 30 cores that we scan multiple times over this experiment to provide a more complete and continuous picture of sound behavior over time. If we look at the day 1 sound speed ratio points, we can see that they're pretty variable and they tend to appear to be higher than the day 30 scans at the same time, given that the rapid elevation change over the first day happened and that the day 30 cores were actually scanned like several hours after the day 1 scans. The day 30 cores may have compacted quite a bit by the time they were scanned. Still, if we look at 2.5 and 5 centimeter depths, which were within the disturbed layer, sound speed ratio appeared to slightly increase from day 3 to day 14, and then slightly decrease from day 14 to day 24, implying this increasing but then decreasing density. Sound speed ratio at 10 centimeters, which was below the disturbed layer, was more variable and changes over time were not as clear, although there appears to be a slight decrease between day 14 and day 24, similar to what was observed in these disturbed layer depths. In this graph, this is the same graph I showed earlier. We have sound speed ratio and porosity values from a wide range of sediment types from other literature. So if we add our average sound speed and porosity values from day 3 to day 30, again, these are the 2.5 and 5 centimeter averages. The differences are pretty small. But if we zoom in, we can actually see that sound speed ratio appeared to slightly increase from day 3 to day 14 as porosity decreased. And then once again, between day 14 and day 30, it looks like sound speed ratio slightly decreased as porosity increased. So these relative changes are also pretty consistent with this broader relationship shown by that black line there between sound speed and porosity. So kind of to bring it all together, it appears that most of the physical compaction occurred rapidly within the first day, but surprisingly, there may have been small density changes after elevation change stabilized that were not apparent or in porosity measurements. And this is likely due to the destructive nature of porosity coring and sectioning, especially for disturbed sediments that might be prone to compaction while you're taking that core. And sediment-water interface height and acoustic measurements are these nice non-destructive measurements that appear to be more effective methods for these small and rapid physical changes. But anyway, let's move on to our cohesive variables. So we expected that changes to cohesion would continue after compaction stabilized. So we measured changes in properties that might integrate the physical and biological contributors to cohesion. So to examine changes in sediment surface cohesion, we measured erosion by exposing cores here to intervals of increasing shear stress from 0.1 to 0.6 pascals using this gust erosion chamber device that Grant and I built instead of paying $20,000 for the proprietary one. So this device uses a spinning disc and a pump that exerts the shear stress on that sediment surface. And then it passes the eroded material through the transmissometer, which is basically a turbidity sensor. So that provides us with turbidity data, and then it's collected in a bucket at the end, which we can then filter to get a value for the total eroded mass at each shear level. So basically we have these two erosion measurements that we expected both would decrease over time with increasing surface cohesion. To measure subsurface cohesion, we measured tensile force with a custom probe that screws into mud. It pulls up and breaks off a mud plug. And so the more cohesive the mud is, the more tensile force would be required to break the sediment. So we expected max tensile force to increase over time. And overall, we expected that surface and subsurface cohesive properties to be related. So we measured EPS, which is that microbial carbohydrate slime, expecting that it would link surface and subsurface cohesion changes that happen on similar timescales. So if we first look at erosion qualitatively, we can see that it appears that overall erosion goes down over time. So this video on the left is a day 1 core at 0.6 pascals, which is the highest shear level we tested. And we have a core at day 7 and a core at day 30 at the same shear stress level. And overall, we just see that the water gets clearer as time goes on. So if we look first at the turbidity sensor data, we also see this decline in the murkiness of water. So at several points during the erosion experiments, turbidity often exceeded the turbidity sensor threshold. So here I've plotted the proportion of time that the murkiness of the water exceeded the threshold at each shear stress level, which are in these colors here, over time. And this demonstrates a steady decline in turbidity over time, especially at shear levels below 0.6, which are these colors under the green bars. And this suggests, as we expected, that increasing surface cohesion continues past the first few days in which physical compaction took place. So we expected the same trend to be apparent in the eroded mass filtered at the end of each shear level over time. However, we see a different pattern. So there appears to be a large drop between day 1 and day 2 in eroded mass, and then it remains relatively low from day 2 through day 14. But by day 30, it appears to increase again, and now is pretty variable as well. So why do we see this discrepancy between our turbidity and eroded mass measurements? So if we go back to what erosion looked like at these different times, we can begin to explain this. On day 1, erosion was mainly in the form of these smaller, loosely aggregated particles, which creates this chocolate milk-like water. Day 7, the murkiness is a little bit lower, but we can still see there's some scour on the surface. That's what those dark patches are there. And then by day 30, the murkiness has really gone down, and we really didn't see much scour on the surface, but there's now a lot of mass being picked up. And this time, much of that eroded mass is actually in the form of compact worm poop pellets produced by these small capitellid and spionid worms that were actually living in the sediment when we collected it. So combined, these data suggest that there was increasing overall surface cohesion, but increasing pellet production by worms at later time points. So given this apparent increase in surface cohesion based on reduced turbidity and reduced eroded mass until day 30, we would expect this to primarily come from increasing EPS slime stabilizing the sediment. Instead, we see that EPS concentration over time in the top centimeter of the sediment was highly variable, and there really wasn't much change at all over time. So overall, turbidity and eroded mass did not behave the same due to worm activity, changing the erosion from these small loose particles to large compact pellets, and the surface EPS concentration did not really reflect this changing erosion. But let's move on to subsurface cohesion. So here we have our subsurface cohesion metric, which is max tensile force over time at 3, 4, and 7 centimeters. And these are subsurface depths within and below the disturbed layer. So overall, the max force appears to increase with time, starting around day 3, suggesting increasing cohesion that took a few days to get started. And this increase is a little more apparent at 4 and 7 centimeters, although these measurements are highly variable. And the force was also much higher at 7 centimeters, which you can see on that y-axis scale there. And this is likely because of the weight of the overlying sediment at this depth. If we look at EPS concentrations at those same depths, we again see high variability and not a whole lot of change over time. So EPS concentration overall didn't really relate to cohesion, but resuspension likely would not have actually changed the amount of EPS present, but rather kind of chopped up the sediment EPS matrix. So how EPS and sediment are linked depends not just on the concentration, but on the microbial and microalgal activity linking these strands of EPS together and to sediment grains. And the infauna also likely affected this EPS structuring through their feeding and mucus production. But combined, these data suggest kind of this overall disturbance recovery scenario in which even small-bodied infauna that are sometimes just an afterthought to a lot of people play an important role in changing sediment structure. So following disturbance, the sediment compacted rapidly over the first couple days and then gradually until stabilizing by about a week. Meanwhile, the sediment density and cohesion more gradually increased until about 2 weeks in, where the density then begins to slightly decrease again while the cohesion continues to increase. And this may potentially result from a lag before disturbed infauna are active enough to increase void space and with their burrows, which would decrease density and increase the eroded mass with their accumulating pellets, but at the same time stimulating more microbial activity and compacting sediment around their burrow walls and overall increasing cohesion. But overall, these data indicate that mud cohesion recovery took longer and occurred more gradually than physical consolidation. Lower density did not necessarily mean lower cohesion, and that temporal changes to surface and subsurface cohesion may be related and mediated even by small burrowing animals. And including biological effects in predictions of sediment reconsolidation is clearly important for sediment transport in dynamic coastal areas. So we still need to look at the individual grain size profiles of these cores to help explain core-to-core variability, which for natural sediments can be especially high. We collected all these, within a few tens of meters of each other, but it can still be pretty high. And we could also further test the infauna effects we observed in an experiment comparing sediment with different types of burrowers and sediment with no animals. So we could look at what the differences between what pellet producers are doing, what deep burrowers are doing, and what near surface burrowers are doing. And finally, I was actually fortunate enough to be able to take samples at several depths offshore right before Hurricane Sally. So sorry, everyone, I may have jinxed us there. But I've been resampling these sites since after Sally and after Zeta to examine whether changes to the sediment properties that I talked about today relate to what animals are there and what they're doing to rebuild their smashed homes. So yeah, I'm still working on processing a lot of that data, but it'll be interesting to see what comes out of that. And with that, I'd be happy to take any questions.
Nice job, Cy. Does anybody have any questions for Cy?
Or is everyone hungry to get to lunch?
I can always ask questions.
Okay,
All
let's
right. Go for it.
Nice job, Clemo, Cy. I'm confused with your name always.
I have too many names. It's okay.
Right. Nice job. So maybe one thing I did not fully get. So all the cores you were working with were intact cores, right?
Yes.
And
they just
so
had.
You will also look in what kind of fauna was in each of these cores to see whether, well, among core variability was a function of density or functional groups or whatever.
Yeah, I would have liked to. Unfortunately, I didn't collect quantitative infauna data on these cores because just due to logistics of the erosion experiments, we weren't able to like sieve cores in between eroding them and then doing the other measurements on them. So it was mostly just at the end when I had the last cores, I had some extra time to kind of see who was in there, and it tended to be these little capitellid worms creating tubes and pellets, and then some paraprionospeo or prionospeo worms probably also. But that's something that I would like to do in the future, is to account for that, because I think, yeah, that would obviously be very important as far as how many things are there producing pellets or making burrows or doing that. So yeah, it's a very good point.
And the resuspension you did like manually, or did you also use some rotating disc and controlled shear stress?
No, I just used this very high-tech method with a turkey baster. And basically I 3D printed a little fork that had holes kind of laterally so that it would send water jets kind of laterally at about 5 centimeters and about 1 centimeter. And we kind of just squeezed it a lot just to kind of mix up the top 5 centimeters roughly, and try to be consistent about, you know, how deep we were resuspending.
Thank you. Good to see you. Good to see you all. I was thinking during the hurricane of you, obviously scared that you would do well, but happy for Cy that he got a hurricane during his thesis.
Yeah, I think I'm the only one who's happy about the hurricane.
Thanks.
Thanks for tuning in, Niels.
I'm pretty happy about it, Cy.
Oh yeah, Brian, of course. Yeah. Sorry.
Does anybody else have any questions for Cy? You know, we ran a little bit late here, so I think people are kind of checking out for lunch. But
totally understand.
Preston, you got a question?
Yeah, hi Cy. That was
a
really nice talk. Good to see you.
Yeah, you too.
Could you go back to the slides where you were kind of showing that level decrease just visually?
Yeah.
And I guess my question is, like, why is there such a big discrepancy from what we kind of think of as like the ground truth? You can see the sediment compacting in that particular measurement, but then none of the other ones seems to track that big trend. What's the...
Well, I think at least for like the water content, I think just the method of subcoring kind of erased that like quick compaction that we saw on day one. And then what could also be happening is when these worms are creating their burrows, they're also making piles of poop. Like the sediment has to go somewhere, and just, you know, measuring the side of the core may not be really reflective of the changing topography. So like Niels actually has looked at changing topography of sediment surfaces. And so that might be a better way of kind of quantifying the total compaction, rather than like looking at the sediment water interface just from kind of points on the side rather than across the whole surface.
Yeah, and can you remind me, where did you have the transducers for the sound speed measurement?
So can you see that picture? It's kind of small, but
yes. Okay. Right. Yeah.
Yeah, so they're basically positioned equidistant. The core is equidistant between them.
Okay,
And
so all of the sound speed measurements were at that kind of depth below the sediment water interface, is that correct?
they were at 2.5, 5, and 10 centimeters.
Okay, yeah, you had multiple,
Yeah.
right.
Mhm.
Okay. Yeah, could you just show that acoustic data slide again real quick?
Sure.
Okay, right, there we go. Okay. Yeah, and what was just in centimeters, what was the total amount of compaction in centimeters?
It went from around like 4 centimeters above to, yeah, kind
Okay.
of, my first measurement was about half an hour after I stopped resuspension. And so, yeah, it kind of went from 4 to less than 1 over the first 24 hours, basically.
Okay, yeah, I guess like so many other acoustic measurements we've all been working on, it's like one effect you think is going to be really big, but it's maybe there's a competing effect that you're not sure what that is, but it's coming into play and preventing your acoustic data from like doing what you might really expect. So I guess more uncertainty.
Yeah.
I would have expected to see a bigger effect in the acoustic data than you saw, and maybe you did too. Anyway, okay, cool. Thanks a lot.
Yeah, thanks for tuning in.
You bet.
Yeah, so thanks everybody for coming, and we'll wrap up and sign off.
Yeah.